So this is video part 2 on genetic recombination and recombinant gene mapping. Again, like I said before, complicated topics. We're splitting it up into 3 videos to try and make it a little bit more manageable for you guys. So in the last video, I kind of went over the underlying principles, crossover, the recombination frequency, why this all works. And this is now we're going to go over an example problem. So this first video, I'm going to kind of cover how you determine an order, a gene order, and then in a later video we'll actually go ahead and do some math and calculate a recombination frequency and build a chromosome map of our own. So consider the linked alleles Y for yellow body, CT for cut wings, and EC for echinus eyes, in Drosophila, the fruit fly. So the following data represents the progeny of a cross between 2 parents of unknown genotypes. What is the order of the genes on the chromosome? So like I said, the first part of this these next two videos is determining gene order. So here's our data. This represents the progeny. Just a little note row here. Remember that plus represents the wild type. So this is the phenotype column. These are all the phenotypes, and this is the genotypes. So if you see something like echinus cut, that states the 2 alleles that are present, that are like observable, and then you can go ahead and assume that the other allele is wild type. So you don't need to say all 3, like you don't need to say like echinus cut wild type. You don't need to state wild type. So this is all the number of individuals with each of these genotypes, and then the phenotypes. Well, we know the genotypes because of the phenotypes we can observe. And then just a quick note again, if these genes were not linked, there would be an equal chance of crossover happening between each gene. So there would therefore be equal numbers of each genotype or phenotype in the progeny, the offspring progeny. So therefore, because there's not relatively equal numbers, we know that these genes are linked and something fishy is going on. Because, like, for example, why are there so few of these and so many of these? So we know that they're linked, and some genes are close together and others are farther apart. Okay, so determining gene order, the first step you're going to take is you're going to go ahead and you're going to figure out which is the parental genotype and what is the double crossover genotype. And you can tell this by basically looking at which is the most common genotype in the progeny and which is the least common in the progeny. And that'll go ahead and tell you what is the double crossover product and what are the parental genomes. And this is just, there's 200, we're going to use this later. But so looking at these, once you've identified them, you go ahead and you just look at what's the difference, what changed between these 2 phenotypes. So the parental genotypes we now know are wild type for all 3 at all 3 loci, and also yellow, cut wings, and echinus eyes. Those are the 2. So one of the parent had yellow wings, cut wings, and echinus eyes, and the other one was wild type in all 3 of those factors. And then you can go ahead and compare that to down here, the double crossover product, and by seeing what changed, you know what's in the middle. And in it, I'll have a slide in a minute that'll hopefully clarify why that is. But so let's go ahead and compare. So we have wild type, wild type, wild type, wild type, wild type, echinus eyes. So between this one and this one, echinus eyes is the one that changed. That's the allele that changed. And then let's look at the other one. We have yellow body, cut wings, echinus eyes, yellow body, cut wings, wild type. So again, where the echinus eyes should be, there's now a wild type allele. So that's where it changed. So now we can go ahead and say pretty confidently that echinus eyes is going to be the gene that is in the middle on the chromosome. And why is that? So this just goes back. This is the same slide right out of that first video, showing you how to hopefully think about crossover. So remember, if we start here, we're going to assume that crossover is going to happen right here. So we're pretending crossover is happening here. In order to track that, we would go the progeny chromosome would look like this. It would go A, B, B, C, and then remember crossover happened here, so it looped over and it has a wild type allele for its 3rd allele instead of the CD because it exchanged information with parent 2. And then the other offspring would have wild type, and a crossover CD. So that's what we have here. This is just showing you what the offspring chromosome would look like. So we're going to go ahead and now we're going to look at a double crossover example. So this should hopefully explain why we know that echinus eyes are in the middle. Because when double crossover happens, we're going to start here. So parent one, we're going to assume that crossover happens. It's a double crossover, which means it happens here and here. It's a double crossover. So right here, it would go AB crossover plus, or wild type, sorry. So AB, wild type, and then crossover happens again, and it comes back over here. And CD is the third allele. So this would be what one of the offspring's chromosome would look like. It would be AB plus CD with this wild type coming from the other parental chromosome. And then the same thing we can start on parent two. It'd be wild type, crossover happens here, BC, crossover happens again, plus, or wild type, I keep doing that. So therefore we know that whichever one changed, if we go back here, whichever one changed between this one and this one, we know has to be in the middle, because remember, crossover is going to happen and it's going to flip that middle gene onto the other chromosome. So whatever changed between the parental genotype and the offspring, we know that has to be the gene that's in the middle. So hopefully that makes sense. So now that we know the order of the genes, we can draw a hypothetical chromosome map, and then we can perform some basic calculations to figure out basically the chances, the probability that crossover would happen between each of these genes. And based on those frequencies or probabilities, we can figure out the relative distance between each of these genes. So we know now, based off that ecnus Is is the allele that's in the middle on the chromosome, but we don't know if this is actually correct, if this is actually representative of how far they are. This ecnus Is could be a lot closer to Y, we don't really know. So we're going to go ahead in the next video, we'll cover out how you figure out how far these genes are from each other. That's it, guys. Thank you very much for watching. The third video should hopefully be pretty accessible. But this is brought to you by Academic Success Center. Feel free to stay connected. Here's all the contact information. And again, my email's on the first slide. Don't be afraid to reach out. Thank you, guys. See you soon.